Johannes Krause was reading a sequencing result in Leipzig in the spring of 2010 when he stopped typing. Krause, a paleogeneticist at the Max Planck Institute for Evolutionary Anthropology, who had spent 4 years developing extraction protocols for ancient DNA from fragmentary bone material, had been handed a sample unlike anything his team had processed before. Not because the sample was large, it was a fragment of a finger bone, the distal phalanx, the tip, small enough to sit on a fingernail. Not because it was old, it dated to between 30,000 and 50,000 years ago, which was well within the range his laboratory regularly worked with. He stopped because the mitochondrial DNA inside it did not match anything in the reference database. Not modern humans, not Neanderthals, not any primate species with a sequenced genome. The sequence was human enough to confirm it came from a member of the genus Homo, but it was different enough from every known Homo lineage to mean only one thing. There was a third kind of human, and nobody had known it existed. The cave where the finger bone was found, Denisova Cave, in the Altai Mountains of Siberia, had been excavated off and on since the 1970s. Archaeologists had cataloged tools, animal bones, evidence of fire use, and occasional human remains without finding anything that changed the established picture of who had lived in that region during the Pleistocene. The site was productive. It was not considered exceptional. Hold one specific detail: the finger bone belonged to a child, a young girl, based on the developmental stage of the bone. We will come back to her in about 8 minutes, and what the full genome eventually revealed about her family will make that detail matter in a way it cannot yet. The object at the center of this story is easy to underestimate: a fragment of distal phalanx. If you laid it on a table, you might not notice it. It is not a skull. It is not a complete skeleton. It does not have the visual weight of an archaeological discovery that changes the field. And yet, by the time the analysis of the DNA inside it was published in the journal Nature in December 2010, it had done exactly that: changed the field, in a way that most major fossil discoveries take decades to accomplish, and some never do. To understand why a finger bone accomplished this, you need to understand what the cave it came from actually is. Denisova Cave sits at approximately 51 degrees north latitude in the Altai region of southern Siberia, close to where the borders of Russia, Mongolia, China, and Kazakhstan converge. The Altai Mountains are one of the coldest consistently inhabited landscapes on Earth. The cave's internal temperature has remained stable at approximately 0 degrees Celsius for hundreds of thousands of years. 0 degrees Celsius is, for the purposes of ancient DNA preservation, almost ideal. Cold slows the chemical degradation of nucleic acids. Cold is why mammoths pulled from Siberian permafrost still have intact cells. Cold is why Denisova Cave, which has been occupied by humans and animals across an enormous span of time, turned out to contain some of the best preserved ancient genetic material ever recovered from a Pleistocene site. The finger bone arrived at Krause's laboratory, having already survived between 30,000 and 50,000 years inside that cold. What the cold had done to the DNA inside it was not destroy it. It had kept it. The mitochondrial DNA, the genetic material inherited only from the mother, passed from mother to child in an unbroken line, used by geneticists to trace maternal ancestry, was extracted first because mitochondrial DNA is more abundant in cells than nuclear DNA, and therefore more likely to survive in ancient samples. When Krause's team sequenced it and ran it against the reference database, the result was specific and measurable. The Denisova fingerbone's mitochondrial DNA differed from modern human mitochondrial DNA by approximately 385 base pairs. Base pairs are the individual chemical letters of the genetic code. 385 differences sounds like a small number until you compare it to the reference. Modern humans and Neanderthals differ by an average of 202 base pairs in their mitochondrial DNA. The finger bone was roughly twice as genetically distant from modern humans as Neanderthals were. That figure, 385, was not a classification. It was a signal. It meant that the population this individual belonged to had diverged from the lineage that would eventually produce both modern humans and Neanderthals approximately 1 million years ago. Not 300,000 years ago, which is roughly when Neanderthals diverged from the modern human line. 1 million years ago, whatever had lived in Denisova Cave alongside or before the Neanderthals had been on a separate evolutionary path for twice as long. This finding needed nuclear DNA to be confirmed. Mitochondrial DNA carries only a fraction of a genome's full information, and unusual mitochondrial results can sometimes be explained by factors other than a separate species. The nuclear genome took longer to extract and sequence. When it came back, it confirmed the mitochondrial finding and added something the mitochondrial analysis could not have shown. The Denisovans, the name the Max Planck team assigned to the new population after the cave, were real. They were distinct, and they had interbred with modern humans. This is the finding that reframed the finger bone entirely. The nuclear genome analysis, completed over the following 2 years and published in a series of papers culminating in a comprehensive study in 2012, showed that living populations in Melanesia, the island region encompassing Papua New Guinea, the Solomon Islands, Vanuatu, and parts of Fiji, carry between 4 and 6% Denisovan ancestry in their genomes. Melanesian populations carry this ancestry at roughly twice the rate that non-African modern humans carry Neanderthal ancestry. This means the contact between modern humans and Denisovans was not a marginal or accidental encounter. It was sustained, geographically extensive, and genetically productive. The children of these encounters survived and had children of their own, and those children's descendants are living people today. The problem with this finding, which became apparent immediately and has not been fully resolved since, is geographic. Denisova Cave is in Siberia. Melanesia is in the southwestern Pacific, more than 6,000 kilometers away. The modern human populations that show the highest Denisovan admixture are not in Siberia or anywhere near it. They are in one of the most remote island regions on the planet. If the Denisovans were present primarily in Siberia, how did their genetic signature end up concentrated in Melanesia, while being largely absent in populations that geographically should have been closer to the source? The answer the field arrived at was uncomfortable in its implications. The Denisovans were not primarily a Siberian population. Denisova Cave was a single point of contact, a place where their range happened to overlap with the cold highland environment, where ancient DNA preserves well enough to recover. Their actual range may have extended across an enormous portion of Asia, from the Altai Mountains south and east, through mainland Southeast Asia, and into the island chains of the Pacific. They were not a relic population huddled in one cave. They were a geographically extensive human lineage whose range dwarfed the evidence we have recovered. The finger bone from the cave was not a representative sample of where Denisovans lived. It was an accident of preservation. Then a second bone arrived, and the story became considerably stranger. A molar tooth, found in the same sediment layer of Denisova Cave, was analyzed independently and found to carry Denisovan DNA. A second molar tooth, from a different sediment layer representing a different time period, was also Denisovan. And then, in 2016, a fragment of long bone, the shaft of a large limb bone, was found in Denisova Cave's deepest excavation layers, in a deposit dating to between 50,000 and 100,000 years ago. It was Denisovan too, but the limb bone fragment also carried something else. Nuclear DNA analysis showed that this individual, a female, based on the genomic evidence, had a Neanderthal mother and a Denisovan father. She was a first-generation hybrid. Her mother had been a Neanderthal, her father had been a Denisovan, and she had lived and died in that cave, in the Altai Mountains, at a time when both species were present in the region. She was given an informal name by the research team, Denny, a first-generation hybrid between two species that were themselves already distinct from modern humans, recovered from a cave in Siberia, identifiable only because the cold had kept her DNA intact for somewhere between 50,000 and 100,000 years. Reading through the Denny paper the first time, I set it down for a while. Not because the finding was complicated, it was actually straightforwardly documented, but because what it implied about daily life in the Altai Mountains during the Pleistocene was not easy to absorb. Two populations that had diverged from each other approximately 400,000 years earlier were still meeting, still producing children, still sharing territory in ways intimate enough to make a family. Denny was not a metaphor. She was a person. Her bones were in a cave. Her DNA was in a Leipzig laboratory. The question that Denny opened was more specific and more disorienting than the one the original fingerbone had produced. The original question was, what species left this DNA? Answered, a previously unknown human lineage, the Denisovans, diverged approximately 1 million years ago from the common ancestor of modern humans and Neanderthals. Denny's question was, If a Neanderthal mother and a Denisovan father could produce a viable child in the Altai Mountains between 50,000 and 100,000 years ago, how often was this happening, and across how wide a geographic range? And if the answer to that is frequently enough to leave a measurable genetic signal in living populations, then the picture of Pleistocene Asia as a landscape of separated, isolated human lineages is wrong. The reality was something considerably more complicated: multiple human species, overlapping territories, ongoing interbreeding, genetic exchange, a world in which the boundaries between humankinds were permeable in ways the textbook model never anticipated. The Denisovan admixture signal in living Melanesian populations tells us that this exchange was not limited to the Altai. It happened in Asia, probably in multiple locations, across a span of tens of thousands of years. The specific admixture events that contributed to modern Melanesian genomes have been modeled as occurring approximately 44,000 to 54,000 years ago, based on the length and distribution of Denisovan-derived genomic segments in living individuals. Those segments get shorter with each generation as chromosomes are shuffled during reproduction. Their current length in living people implies roughly how many generations have passed since the admixture occurred, and therefore roughly when the meetings happened. 44,000 to 54,000 years ago, somewhere in Southeast Asia or its adjacent islands, modern humans met Denisovans and produced children who survived. We do not know what language was spoken, if any. We do not know whether the meetings were peaceful or coerced. We do not know whether the children were accepted or marginal. We know only that they lived long enough to have children of their own, and that those children's descendants are alive and living in the Pacific today. The cave in the Altai Mountains is still being excavated. New sediment layers are opened each field season. In 2019, a Denisovan jaw fragment, not from Denisova Cave, but from Baishia Karst Cave on the Tibetan Plateau, at an altitude of 3,280 meters, was confirmed through ancient protein analysis by a team that included Frido Welker, a paleoproteomics researcher at the University of Copenhagen who had spent 6 years developing mass spectrometry methods for recovering protein sequences from ancient bone when DNA preservation had failed entirely. The Jiahi mandible, as it is called, dated to at least 160,000 years ago and demonstrated that Denisovans had occupied the Tibetan Plateau at high altitude long before modern humans were documented there. The Tibet finding raised the same question the Melanesian admixture data raised, but from a different direction. What physiological adaptations did a population need to survive at 3,280 meters altitude for 160,000 years? The answer, it turns out, was already known. A variant of a gene called EPAS1, which regulates how the body produces red blood cells in response to low oxygen, is present in Tibetan populations today at very high frequency and allows them to live at high altitude without the negative health effects that affect lowland populations. That EPAS1 variant is Denisovan in origin. It entered the Tibetan gene pool through admixture with Denisovans, and it has been under strong positive selection, meaning it was so advantageous that people who carried it had more surviving children, generation after generation, since modern humans first moved into the Tibetan Plateau. The Denisovans did not just coexist with modern humans. In at least 2 documented cases, the immune system variants in non-African populations generally, and the EPAS1 variant in Tibetan populations specifically, they equipped modern humans with adaptations that those populations could not have developed on their own fast enough to survive the environments they were entering. Without Denisovan DNA, the Tibetan Plateau may not have been permanently habitable by modern humans at all. The finger bone that Johannes Krause was reading in Leipzig in 2010 has been transferred to the permanent collection of the Max Planck Institute. It is kept in controlled conditions. It is approximately the size of the tip of an adult index finger. From that object, and from the sediment layers surrounding where it was found, the field has recovered the complete mitochondrial and nuclear genome of a new human species, the genome of a Neanderthal-Denisovan hybrid individual, the identification of Denisovan occupation of the Tibetan Plateau at 160,000 years before the present, the discovery that 4 to 6% of Melanesian ancestry derives from Denisovan interbreeding, and the identification of at least 2 functional genetic contributions, immune variants and the EPAS1 altitude adaptation, that Denisovans passed to modern human populations where they remain in active use today. None of that was visible in the bone. It was visible only in what the cold had preserved inside it, and only to a laboratory with the tools to read it. What the field still cannot answer is the question the Denny finding sharpened. If a Neanderthal mother and a Denisovan father could meet in one cave in Siberia and produce a daughter who survived long enough to leave bones, if Denisovans ranged from the Tibetan Plateau to the islands of the Pacific, if their genetic contributions persisted and were selected for in populations living today, then how many other Denisovan individuals are sitting in caves and riverbeds and lakeshores across Asia in conditions where the DNA did not survive, in countries where the excavation has not yet happened, carrying evidence we have not yet found the tools or the opportunity to read? The finger bone was not the whole story. It may not even be the beginning of it.